Thank you. Good afternoon, everyone. My name is Poonam Mathur, and thank you to the organizers for inviting me to present our data. It's my first time at this conference, and I'm learning so much and really enjoying it. I think our data falls at a good time after Dr. Rabi's presentation because I may be able to introduce a concept to you that goes beyond statins for intervening on the hyperinflammatory state in women living with HIV. So I don't have any disclosures, and I'll provide a brief background. So we know that the life expectancy for people with HIV has now approached that of the general population in the United States, where people with HIV live to an average of 74 years, and the general population, meaning those individuals without HIV, live to an average of 80 years, and this is largely due to effective antiretroviral therapy. And concomitantly, we've seen a decline in mortality from HIV, especially among women. So since the 1990s, the mortality in women living with HIV has decreased dramatically. However, the morbidity in women living with HIV has not decreased. So this is data from the multi-center AIDS cohort study and the Women's Interagency HIV study showing that with every increasing decade after 40 years of age, women living with HIV have more non-AIDS related comorbidities than men living with HIV, despite viral suppression from antiretroviral therapy. So as Dr. Rabi was mentioning, part of the reason for this increased rate of non-AIDS related comorbidities is due to the hyperactivation found in women living with HIV. So what this graph is showing, and I'm sorry I don't have a pointer, but on the left-hand side is showing that with uncontrolled viral replication, there's immune activation and inflammation that ensues. Then the decline that you see, the drastic decline, is due to the advent of ART and viral suppression. But as you see, the immune activation from HIV never completely goes away. That's because of the HIV reservoir. So this inflammation continues, and then with biologic aging, there's an exacerbation of this inflammation that's in the body. And that contributes to what's highlighted, which is the HIV aging phenotype. So this phenotype is characterized by those non-AIDS comorbidities, including cardiovascular disease and frailty. So it's important to determine if there's a cellular mechanism or an immunologic mechanism that can be targeted to subdue this inflammation that's found in women living with HIV. So one thing we can look at from an immunologic perspective is this marker called CD38. So CD38 is not new in the field of HIV. It's found to be increased on T cells after HIV infection. And what was found in the multi-center AIDS cohort study is that among people with HIV who were infected for at least 8 years, CD38 expression was a prognostic marker for the development of AIDS within 3 years, and was also a marker for morbidity and mortality. So in my group, what we did was investigate CD38 a bit further to see if we could link hyperinflammation in women living with HIV with CD38. So I'll show you some background data. This is what we've published so far. So we looked at people with HIV on and off ART and compared CD38 expression in those groups to people who do not have HIV. So as expected, people with HIV with viral replication had high levels of CD38 expression. It decreased with ART and viral suppression, but did not normalize to individuals who do not have HIV. So there's persistent CD38 expression despite suppressing viral replication. We also found that women living with HIV who are virally suppressed have persistent CD38 expression that is increased compared to men living with HIV who are virally suppressed, and these are age-matched. So we have persistent CD38 expression, which is much higher in women living with HIV. So since CD38 was found to be important in HIV, it's also been found to play other roles, and one of them is that of an enzyme. So it degrades intracellular nicotinamide adenine dinucleotide, and if you reach back into your brain to some chemistry class or biology class, NAD plays a crucial role in mitochondrial function and cellular metabolism. So CD38 degrades intracellular NAD, and we confirmed this relationship in our lab. We looked at CD4 and CD8 T cells and cultured them with the CD38 inhibitor called 78C. So you can see after CD38 inhibition, NAD went up significantly. So there's this inverse relationship implicating CD38 in mitochondrial dysfunction due to NAD depletion. Next, we looked to see whether CD38 plays a role in inflammation. So we looked at production of inflammatory cytokines. On the top, interferon gamma, on the bottom, TNF alpha. So what we did was stimulate CD8 T cells with HIV GAG peptide. GAG is very important in HIV for viral machinery and basically helps the virus infect. So when we took CD8 T cells and stimulated it with GAG, not unexpectedly, the production of inflammatory cytokines went up. And then what we looked at were the proportion of cells that express CD38. So among those cells that produce inflammatory cytokine, more of them also express CD38. To further confirm the relationship, we cultured the cells with the CD38 inhibitor 78C after we stimulated them with GAG peptide and found that the inflammatory cytokine dropped significantly. So this suggests that CD38 plays a crucial role in the pro-inflammatory state that's occurring in people with HIV. So this brought us to our research question. Are there differences in CD38-induced inflammatory cytokine production between women and men living with HIV that could account for the increased rates of the HIV aging phenotype in women? And then we wanted to see if there were variances in the genetics between women and men living with HIV to account for the differences. So we took samples from people with HIV and the New York Healthy Donor Cohort. So we had 6 people with HIV and 6 individuals without HIV. There were no significant differences in the mean ages between the two groups, and most people were Black. So we looked first at our cytokine production. We stimulated peripheral blood mononuclear cells, which are basically immune cells, with the generic stimulus. Those were anti-CD3, CD28 antibodies. And we looked at the proportion of cells that secreted the inflammatory cytokine interferon gamma and measured how many of those cells also express CD38. Next, we looked at our RNA sequencing to look for genetic variances. So the top flow chart just describes the different steps we took to find differences in gene signatures between men and women living with HIV. So we did some quality control measures that are listed there. And then what we did was looked for differences in gene expression by looking at the genes that had at least a 2-fold change between CD38 positive cells and CD38 negative cells. So first, what I'll tell you about is the inflammatory cytokine production. So what's being shown here by the colors is just differences to sort out the cells using flow cytometry. And on the right-hand side are the results of differences in inflammatory cytokine production. So what we found was that in women living with HIV, there were more cells that produced interferon gamma that expressed CD38. But that difference was not seen in men living with HIV. So it does seem that CD38 plays a different role for inflammatory cytokine production in the women than men living with HIV. Next, we looked for differences in the gene signature. So what this is showing are like heat maps for each of the 4 groups. And it's just a broad overview. It doesn't describe anything about individual genes. But what you're looking at is that if you have more of a red color, you have genes that are upregulated, and the ones that are more blue are genes that are downregulated. The first and third groups are the people with HIV. The first group is men with HIV. Third group is women with HIV. So just as a broad overview, you can see that amongst those groups, the CD38 positive cells have a different gene signature than the CD38 negative cells. So now I'll tell you about the different gene pathways that we looked at. So the first group were the genes that coded for inflammation and cytokine signaling. So the genes that we found are listed in the second bullet point. But what we found, again, not unexpectedly, was that the genes for inflammation and cytokine signaling were all upregulated in the CD38 positive cells for all people with HIV. So again, on the right-hand side is a close-up of the heat map. So when you see more of the red color, that means the genes are upregulated in those groups. Next, we looked at the pathways for mitochondrial dysfunction. Again, the genes in that pathway are in the second bullet point. So what we found was that for mitochondrial dysfunction, the genes that are associated with that were upregulated relatively in the CD38 positive cells for all people with HIV, but even more so in women living with HIV. So implicating that maybe mitochondrial dysfunction is involved in hyperinflammation in women living with HIV. And last, we look at genes associated with the NAD signaling pathway. So what we found were that the genes were downregulated for NAD signaling in the CD38 positive cells of women living with HIV compared to men living with HIV. So this is really important because we've seen that women living with HIV have higher proportions of CD38 expressing cells, and we know CD38 degrades NAD. So now it seems like there may be a genetic signal for the downstream effects of NAD and the lack of NAD signaling that could lead to hyperinflammation in women living with HIV. So in conclusion, we know that CD38 expression persists in people with HIV. This is despite viral suppression by ART, and it's increased in women living with HIV and associated with inflammatory cytokine production in that population. We know that the NAD signaling pathway based off of genetic variants is downregulated in the CD38 positive cells of women living with HIV, so this may mediate increased inflammatory cytokine production in women with HIV. And so this CD38 NAD axis may be a mediator for hyperinflammation and a potential therapeutic target to mitigate inflammation in women living with HIV. And the hope would be that we can translate what we know about this axis to develop potential therapeutics that could serve as adjuncts to ART and hopefully reduce the risk of end organ disease. So I'd just like to take a moment to acknowledge my co-investigators, Drs. Pozniak and Ghosh, and my mentor, Dr. Kodalil. And of course, I'd like to thank all of our patients who volunteer their time to participate in our research. Thank you.
Thank you very